think we'll quickly reconvene. The topic of personalized medicine is sort of high on everybody's radar screen these days, and we are moving in many areas to a more personalized approach to care. And as the human genome now, then data about that becomes more available to everybody, influencing how we practice medicine and what the role of those things are going to be is very timely and important, because it's changing how we care for people, and hopefully will make how we care for people better. So Dr. Greenhalgh is going to give us an introduction.
Thank you very much, Dr. Farmer. Dr. Bold asked me to give this talk at American College of Surgery last year, so I brought it back for you guys, and I have nothing to disclose except that you might hear some basic science, so I'm sorry about that. But, what's the goal? Can you target your treatment based on the genome, the gene structure of your patient? So you have to determine what their structure is and kind of design the treatment. So I'm going to go over how we got involved with this using the glucocorticoid receptor gene as an example. This is from our laboratory and working with my lab gang. So, the question I've had for many years is why is there such a different response to inflammation? I think two brothers with the same burn. One does extremely well. One gets sick, it's septic, doesn't take a skin graft, ends up dying. We can never figure out why one does well, one doesn't. So, and we deal with the most severe stress, so it's a good area for investigation. So, what's the story on steroids? Well, studies are around, you've seen them all, that first they were bad, then they're good, and they can help. You can test for steroid response, and the latest studies say, no, there is no benefit. But it still comes out to, if you look at the Surviving Sepsis campaign from 2013, that, all right, you guys, do everything you can to stabilize the patient, and if all that fails, give steroids anyway. So we still give it as a rescue, so we need to know more of what's going on. So we also know that a lot of times we treat people for diseases with steroids, and then there's an incidence of resistance to steroids, 10% nephrotic syndrome, 1 in 1,000 asthma, inflammatory bowel disease, 15 to 20%. So we need to know why they might have this resistance. So the questions I'm asking, why are there differential responses to stress, and same issue, and why are there resistance to steroids? So we'll go into that. Now, if you think about gene expression, all the cells have the same genes, and so the neurons may not want to respond to steroids, but they still have the gene, but you maybe want the inflammatory cells to. So what regulates that response? Why do some cells respond, some don't? And so we're going to talk more about that. So one of the secrets in research is check your data carefully, okay? So, we are working now, I guess it's about 10, 15 years, 12 years ago maybe. And so we're doing a study, we're looking at the glucocorticoid receptor, and back then we're looking at the difference between a normal mouse and a mouse that had CD14 knockout, which is related to LPS signaling and all that stuff. And we saw differences. You can see here that, oh yeah, in the knockouts you don't respond as well. So what? But Kehoe chose, the PhD I've worked with for many years, only 17. He noticed, look at this. This is different size, and you see a little tiny difference in the size of this plot. So what do we do in the lab? We take it, we sequence it, and then we found out that, wow, there's a difference in that glucocorticoid receptor. So it's kind of tedious, but in the normal control, there are these things called CAG repeats, which give you a glutamine, what they call it's poly-Q, and poly-Q causes diseases. And what we found was that the knockout mice only had 8 of them, but the normal control mice had 17 of them. So, all right, well, so what? So what we do, we test to see if they had a differential response. And what we found was that we could take and put as many of those CAGs in the gene and look at the differential response. And here we have down here is where the knockout mice were, and this is where the controls were. Well, we also noted that the C57 black mice have much better resistance to LPS treatment than the knockout animals. So maybe the reason is that they can handle stress better because they have a better response to steroids. So, all right, so you work with mice, but let's go to the people now. So we hypothesize that people have different, basically, gene structures, that maybe that's why they have a differential response to burns, trauma. And so, of course, we go ahead and do it. Now, I'm just going to, sorry about this, but the glucocorticoid gene is like this. It's got basically 9 exons. The exon 1 doesn't have much function, but the real coding region is here, and then you can either have the 9 alpha, which is the functional, or the 9 beta. And it turns out that you can have different forms of it, and this is the structure. You have a transactivation part, DNA binding domain, hinge area, and a ligand binding domain. Okay, so fine, you know. And also, I'll just point out that there's also the RNA has a 3' end and a 5' end, and we'll talk about that in a little bit. So this is what happens normally for the steroids to reduce inflammation. You have the steroid bound by serum binding proteins. They're released, they go to the receptor. Now all these receptors have chaperones that limit its activity, then they dimerize, then they go to the gene, and there's a GRE, or a glucocorticoid response element, and then you produce your anti-inflammatory responses. And it's sort of simple, but it's actually more complicated than that because sometimes it doesn't dimerize. But anyways, that's the simple process. So what do we do in our lab? We draw blood from people. The universal donor is whoever we can find in the lab. And then we take it and we sequence what we find by doing RT-PCR. And we look at basically the blots, and we say, all right, we have different ones, and we sequence them, and we do a functional assay. And that's kind of the basics of it. And here you go, and it turns out, just an example of several people, they had differential responses. So in differential sizes, so there are variations in the structure of this gene. And what we found was that some had early termination of parts of it, some had, well, some people are the normal. There is a normal, you know, when you sequence it and you can go to the NCBI and you can find out this is the normal sequence. Well, that's crap, that's just one person. Everyone else has different genes, so they change based on different people. Actually, we found 11 people with just that reference size, and everyone else had different sizes. So here's an example. We found a bunch of people. Here's the normal. We found one person. But all these people had little stop codons. They had variations that would lead to small fragments. And not all of their, sometimes they have the normal one and this. And of course, you test a protein that's been truncated. It doesn't have much activity, right? So, and we showed that. That's not too exciting. And then we found this person, patient, or subject 66, and so we found their thing. And they also had a few other components in addition to the normal gene. And so we tested them, and it turned out, now this is what the normal response is. We found a person who has a super response, 6 times the normal range. Now this has been documented for a lot of people that if they have a super response, they frequently have obesity, hypertension, and some other metabolic changes. So we found someone, we don't know who that person is, really, and we could see that this is a super responder. Now it turns out that this one gene has 3 mutations where there's just one switch in the base pair in each one of these 3 sites. So we could actually take and say, all right, let's make the glucocorticoid receptor and make basically single mutations. And this is a work by one of your former co-residents, Kelly Tong. And we can make that gene, and then we can also combine 2 of them in every combination. We can see what combination gives you the best function. It turns out you need to have all 3 mutations in order to show that, hey, wow, that's the optimal response. Now, there are even people, there's a guy at University of Oregon who's shown that he could go back to an evolution and show that how these mutations led to the origin of the glucocorticoid response. So you can go through the whole thing, and a single mutation or a double mutation may lead to some hyperactivity, but otherwise no. So we did all this, and that was interesting. And we found a few other mutations that led to hyper response. This work done by Dr. Lasker showed that, okay, we had one mutation, it's a hyperresponder at baseline. But you know, you want to know whether the receptor responds in response to the dose of steroid that's released, because normally you don't want to have a response, but if you're stressed, your steroid levels go up, so maybe you want to have a super response. So we did a dose response, and it showed that these two guys, especially the yellow one, that mutation, is a better responder to stress at higher levels of steroids. So that work is just about to be published, right, Michael? So, all right. We also looked at preliminary evidence to see if cytokine expression would change. We saw some preliminary studies that there is a functional difference based on these different areas. We can actually block the receptor using RU486. That's the after fooling around drug that stops pregnancies. It blocks steroids, and it blocks the whole thing too. All right, so that's interesting. So you can actually go and you can say, all right, I'm going to treat you, but I want to look at your structure of your steroid receptor, and maybe I can find out whether you need a different dose or not. So that's what we did in vitro, and we did find interesting. You see these little extra little bands, okay? And so we went through and said, all right, let's look at some of these extra bands in more detail, okay? And so you do that, and here's that band, and you can look at one structure. Well, this is a shortened version. Normally you have 777 amino acids in your glucocorticoid receptor. This one's mutated. Then we found this silly one here that's got a stop code on one loss of a base pair, and it stops at 118 amino acids. So clearly that one won't work, so we tested again, and of course it doesn't work. However, we did a dose response curve. Okay, now this area down here is what your normal response is to steroids. And by studying that little fragment, 118 base pair, we got a super response. So wait a minute, how does that work? So we found this multiple times. We've published this work now. Aaron Baker, who just left, did a lot of this work. We also saw that also with methylprednisolone. And so it seems like even though normally you have all these parts, all this 118 base pair has, it doesn't have the DNA binding domain, it doesn't have a ligand binding domain. We don't know how that works. But it turns out, in a lot of times in nature, that genes may express a normal large-sized protein but make a subfragment that either inhibits or augments the activity of that gene. So we may have found a cofactor that augments the response when you need it during times of stress. So it's not just the gene, you know, so it's they and all our cells. And we know that the gene, what turns it on, what turns it off. So there are things like DNA methylation. Now, if you have a methyl group on part of the gene, it shuts it off. So we know that. So if you take off the methyl group, it'll be active. We have all this miRNA. So you have RNA interference microRNAs, and they can be part of the gene, and they can stop it from being expressed. If you block the miRNA, and maybe it's active. And there's also parts like we're looking at the 3' UTR. The UTR stands for untranslated region, means it's just part of the RNA, but does it influence how the gene is expressed? So this is the simple version. And here's down here, the gene's there, but there's a 5' UTR. That's the promoter. So the promoter may be active to tell you to go ahead and be active. And then there's the 3' UTR, which also is where the miRNAs may inhibit the gene activity. So these things actually may regulate whether the gene is in one form or another. And this part of the 5' UTR, you may have binding where a certain receptor binds to it and turns it on or turns it off. So it's getting more and more complicated all the time. So I'm going to show you, this is now the current structure of the glucocorticoid receptor, this part up here. Wow, that's the real gene part. This is the promoter region. In the promoter region, or the 5' UTR, actually goes up 35,000 base pairs. And you can see here, that you can't really see it, but there's these binding sites here for the GRE, so you can actually have binding to itself from the steroid receptor to turn it on itself, and AP1 site, so you can have it, all these different signals to say whether you want this structure or that structure, or turn it on, turn it off. And this is the 3' untranslated region, which has all these binding sites for RNA, which may turn it on, turn it off. We also have these polyadenylation sites here. So you think genes are simple? No, they're incredibly complex. And each one of these areas may determine whether you have active, inactive, which structure of the gene you might have. So we went further to say, all right, we have this little 118 amino acid protein. So we know the 3' UTR is there. Let's get rid of it and see if there's any activity. So we did that, and we found out you need to have the 3' untranslated region for this little 118 amino acid area to work. Well, which part of the 3' UTR is important? So we actually, and it's pretty incredible what you can do with genes nowadays. You can just chop up and take out segments basically bit by bit and make it shorter, and we took out 300 base pair segments. And well, you figured as you get rid of more and more of the 3' UTR, it won't function, right? So we did that, and here's an example of, this is the original hyperactive component, and we took out segments, no activity, no activity, no activity. Uh-oh, we got to the minus 1200, all of a sudden super active, even more potent than the 118 base pair. And it turned out at that site, there's a polyadenylation site, which is an area that could be an enhancer for activity. So we've shown more that this is an area that might be hyperactive. And it turns out that this 1200 loss isn't even regulated by the glucocorticoid, isn't even regulated by the dose response. So down here, this is the dose response, the original 118, I'm sorry. But the other one was not. And to make it even more interesting, we did studies with RU486 that blocks the activity. And if you block the steroid activity with RU486, it blocks that 118 amino acid function. So we think it might be a co-receptor with, or a cofactor with the glucocorticoid receptor. So then we started thinking like, well, maybe stress in itself may lead to a variable response to your gene. So maybe normally you had to have a certain structure, but maybe this little 118 amino acid protein is induced by stress. So it might be cool, you need it, you can produce this cofactor, increase the stress. So, dang it, go back. So what we did was we looked at a lot of people, and we found this short fragment in several other people, and went back and looked for it. And we actually were looking at whether the RNA, whether the changing the structure of, or shortening the 3' UTR would change the RNA structure. So you can actually do these cool diagrams looking at the RNA structure. And we saw differences. I don't know how much to interpret this yet, but we're still looking at it. So we followed the poly-A site. And so what we found now is that you could take out fragments of the 3' UTR. You can see hyperactivity or hypoactivity. And the RU486 suggests that it has some binding to the normal glucocorticoid receptor. So a lot of polymorphisms exist. And there's early termination forms that inhibit activity, increase activity, and so we look at that. So that's fine. Maybe you can use this kind of strategy for treating people. So if you have a hyperactive glucocorticoid receptor and you need to treat someone with steroids, you can figure that out and say, all right, I'm going to give you a lower dose. And maybe you could do something like this. If this is your structure, we found a dose response based on these, so maybe you'd be better to treat them with dexamethasone. However, if you have this mutation, you're a hyper-responder to methylprednisolone, so you could adjust the steroids based on what your structure and in vitro activity might be. So that's the personalized part of it. Well, that's interesting. That's all in vitro. We took normal people, so you really want to take it to the bedside. So what do? We take it to the bedside. So we're doing a study now. It's ongoing. We have basically looking at burn patients. Burn patients are ideal because they have the most stress of anybody, any kind of trauma. So it's the positive control, you could say. So we want to know, can you get variations in the glucocorticoid receptor, and does it change over time? We're doing Controstin stimulation tests, so give ACTH, so you have the variability. We want to know if they have this truncated form that's hyperactive. So we've done a study is basically patients with burns greater than 20%. It's IRB approved and it's ongoing. We draw blood at different types. We follow their clinical course. We do some ACTH testing. We're looking at how many people are there. We've done now actually 72 patients. And fortunately, I have funding now for 4.5 more years to look at this, so we may have quite a few patients. We are still going through the correlations, and we need to know if the different form is created during times of sepsis, or if someone dies, they have a variable form, so we have to go through it and do that. So just a snippet of what we've found, and we have had 20 out of 32 so far that we've analyzed have no single nucleotide polymorphisms, but we have a lot of people who do have changes. For instance, we have several people who have the well-known variant called HDR gamma. I don't want to go through these in too much detail because it's somewhat tedious, but we found multiple variations in different people. For instance, we've even found that you could say that some of these people develop new exons inside the gene, and you can do that based on, you can find where the splice donor and acceptor sites. So in theory, these are people that could make a different form of the protein. And we found now we've got 6 people who have different exon sites in different introns, and so we're starting to analyze what those might be. Again, I don't want to get too much into detail of how that occurs because it's somewhat boring. But anyways, we'll figure that out more as we go. So we have found that there's variability in patients. We have found that people do develop that small fragment that may be a cofactor that augments the glucocorticoid receptor activity. So we have now started to do the correlation. So we have now been looking at one patient we've had who was a 46-year-old man with 90% burns, and he consented to the study. He was in the hospital for 9 months, and so what we could do is we could draw blood over time. We are starting to do the clinical correlations, but just to give you a sample here, here are different samples over time. Now these are all things, they always have the baseline glucocorticoid receptor, but if you really look closely, they have all these changes in these additional forms. And during different periods, that may mean that he has either an augmentation or a decrease in the receptor response. And one of our theories is that stress may induce a variation in the gene so you can handle that stress better. And you can see the variability. And we've actually started to look at, here's IL-1 beta. Yeah, there's variability. We need to correlate it. We did find at one period now, here we are about 32 days out, a huge increase in the COX-2 expression. So maybe at that point, and again, we have to go back, so maybe in a couple of years, we can go back and say, we have found that during periods of stress, these cytokines are hyper-induced in response to the stress of the injury. So clearly, he's got variations over time, and as we do more patients, they may vary over time. We need to do the clinical correlations. And our theory is that the HDR expression appears to be variable based on the stress of that situation. So where are we for the future? We want to look at the variance in relation to stress. We want to find how the variants influence mRNA expression. One of the theories might be, is there a signal that tells the promoter that you should give a different form of the gene to give a different protein? We need to look at that. Maybe there's a danger signal, that's what a DAMP is, or a PAMP is a pathogen-associated molecular pattern, such as it's a part of a viral DNA, or maybe part of a bacterial component that goes to a signal, maybe toll-like receptors, maybe another one that says, all right, change what the structure of the glucocorticoid receptor might be. And we want to look at what pathways might be involved in that signaling. And then what are the epigenetic regulators? DNA methylation, siRNA, the 3' UTRs. And we're doing this. Now, you'll hear from Kehoe Cho. Kehoe and I have been working together for 17 years. He's the real brains behind the organization. His quote is that the genome is not static. Your DNA changes, and we know that the DNA changes over time. So there are DNA that jumps back and forth, and that's what we call the triome, transposable retroelement landscape, which means that normally your DNA, there's parts of viruses can insert and change the gene. Your DNA may change over time. If you look at your hypervariable part of the immunoglobulin, it's DNA that jumps back and forth that allows for that variable components to your T cells to function. So your genes, your structure of your DNA changes over time. That's why you guys don't know, I have different color spots as I get older because there's mutations on my skin. And that's also why you get a cancer in the future. That's because there's DNA that changes over time. So it is likely that stressors influence how the genome changes in structure over time. As a matter of fact, we saw recently an NIH grant proposal that proposed the 4D DNA landscape. The 4D, it's got the primary structure, secondary structure, the 3D structure, and the fourth dimension is time, so it changes over time. So that's kind of one of the theories, that your DNA changes over time. So I don't know if you guys remember who Lamarck was. Lamarck's theory of evolution was that what changes happen during your lifetime could be passed on to your children. Well, Darwin kind of poo-pooed that and said it's your DNA that changes. But Lamarck is right to some extent. What happens during your lifetime can affect it. Now, most of the time, your changes occur later, so you have to have the change before you have your children. So that's a lot of times your children come before you get old and get cancers and things like that. But at least he's partially correct. I like this quote, and this is what I'll finish off with. You can't solve problems by using the same kind of thinking we used when we created them. Because when I was starting out, it was the DNA and the gene. We looked at gene expression, we looked at PCR, it's upregulated. So what? No one's really gotten very far from it. So as the DNA now changes, as the gene changes, as you figure out what regulates where the genes turned on, you need new technologies to figure this out. Albert Einstein was a pretty smart guy, I think. So, and I'll just finish off by acknowledging Keiho Cho and I have been partners for 17 years. So if you guys want to go into research, if you can basically partner with a molecular biologist, because I come up with ideas and Keiho carries them out. And by the way, if any of you guys want to come into my lab, you can get work done. Mike Lasker was in the lab for 2 years. Aaron Baker contributed to this work. Michael did. Kelly Tung, Christian Yee, Amir Amini. All people who worked in the lab, we'll pay your way. Dr. Farmer, we pay the way, so you can send them to us. We have an excellent clinical research staff, as you can see listed on the right. And fortunately, I've been funded with the Schreiner grants for almost 25 years now, and for 4 more years. So thank you for your attention. I'd be happy to take any questions. Dr. Farmer.
So Dave, thank you very much. I really can imagine a time when not only do you take the patient's history and physical, but you have their microarray DNA data. And we as patients, well, our primary care docs will get this on us. And you'll know, oh yeah, this guy comes in, he's a responder to this medicine, or he doesn't respond to that medicine, and you better do it. This is what personalized medicine is all about. What I didn't realize is that it wasn't going to be easy. I kind of thought you would just have that blueprint, and you'd have the checkbox that said, oh yeah, this drug, that drug, not for this guy, yes for that guy. Didn't appreciate how much it was going to change over time, and that
it changes over time.
Changes our DNA.
Right.
Is what you're telling me.
So it changes over time. The structure is influenced by other things, and so yeah, and you can get and it's been, people have done gene chips for a long time, and they show, here's our gene chip, and they have this big square with reds and greens, and they change, and so isn't that interesting? But it's never really been used to kind of design the treatment. But maybe, Dr. Bowl can say, all right, the main mutation in your cancer is this. So cancers are usually fairly uniform, although they mutate too. So you can kill 99% of the cancer with the chemotherapy, and then that one mutation goes on. That's why I have trouble treating all of them. But yeah, it's much more complicated than here's a gene, block it. And our sepsis studies, they have tried to block everything, LPS, TNF, IL-1, IL-6, and nothing works because mutations, there's redundancy in the system, so a lot of those things don't really affect how you respond. Tina, I'm Dr. Palmer.
15 years and I'm still trying to figure out what you're saying. As best as I can, we all have a common isoform and we all have variants. And what do you view as the role of the variant? Is it a modulator of the system under normal situations? Does it get activated when there's a stress? Or is it a fine tuner of the system? And what triggers it?
Yes. Well, that's what we're looking to figure out, because I think that's indeed the case. Now, I was going through literature and looking for, and I found other examples where a lot of main proteins will produce a fragment that will lead to variations. Now, if you look at I can't say it, propio, PIOP, whatever, that makes melano MSH and ACTH, the body makes fragments and each one has a different function. It's pretty common for our body to take a gene and make one part that does one activity and breaks it to another part that makes another activity. And the idea that a lot of the genes will produce a little fragment that inhibits its activity, and you may influence whether you make that little fragment. So what we need to find out is what makes a little fragment. And first of all, we see a lot of these people that these little extra bands. Are they really being transcribed into a protein or not? We don't know. Every time we do these studies, we can do a Western blot and show it the actual size of the protein. We need to go now, and we've had a little trouble finding appropriate antibody for the early part of the glucocorticoid receptor, but we've found when we're starting to look at whether we can find the proteins in people. So we need to go look for these little fragments in people. And when we send papers in, one of the critiques is, all right, show me a syndrome related to this problem. So we don't have, it's easy when you're an endocrinologist, you used to have someone who's hypertensive and you find a mutation in their glucocorticoid receptor. That's where a lot of these have come from. But we're hoping that the burn patients will help us during sepsis. This one fragment's always turned on for those who get better, not turned on for those who don't get better. And you're right, those are the questions. What I've found is the more you do research, the more you don't know, and that's kind of what we're seeing here. Rick, Dr. Perez, sorry.
So you're painting a picture of a very complex system. And as you bring more of these pathways into play, it seems like along with the molecular biologist, you need sort of a bioinformatics collaborator to make sense of this. Can you comment on that? How can you, are you working with somebody like that?
Actually, we are. So Dr. Perez asked if you're working, it's so complicated, do you work with a bioinformatics person? And actually, we do have a bioinformatics, a couple of people working with us in collaboration, because what Dr. Chill wants to do is, you've heard of the genome, he's working on the triome, which is the transposable rental element genome. And so to try to create a database of what are the different kinds of mutations that you might find that influence, for instance, retroviral insertion, we call line signs. Lines are long interspersed nuclear inserts. Those are the DNA parts that jump in. There's different components of them. But yeah, it's so complicated that we are collaborating with bioinformatics people. As a matter of fact, Kehoe would be here today, except he had to fly to Korea to try to defend a grant for $5 million to collaborate with people in Korea. So and a lot of that is with the bioinformatics group to create a database. It gets so complicated. And he'll show that he's got fancy little drawings where you can kind of, he takes, shows how parts of DNA coapt, and you get these little diagrams. I think he's shown them before. They look like transistors. And maybe there's a pattern to that. So, to understand what's going on, you need to have the bioinformatics people. So we are collaborating with that group, and it's getting more complicated. And his thing is that, you go to the database, and we've cloned our DNA. Well, that's one person, and really, if you look at it, there's all sorts of missing sections, because when there's all this repeat stuff, there's all these areas they have no idea what's going on. They have sections they know about. But, I don't look like you, or I don't look like her. We all look different, and there's little segments or difference in how we look. And if you think about it, a mouse and a human have very similar genes throughout the body, and what the difference is some of the stuff in between the genes that influences whether you're kind of big and bald, or you're small and hairy, or those kind of things. So it's so much more complicated than it used to be.
But if you think about the ability to apply new technologies to communications that's occurred in just 3 decades, that was the application of big data, and all of the comments about the transistors and how small they have become over time, and how many you can get on a chip, and all of those things. It's sort of amazing that we haven't applied that same kind of advanced medicine. We're essentially doing the same kind of medicine we were doing 30 years ago. Everybody who gets sick gets the same dose of antibiotics, and the same kind of antibiotic. And that the fact that we haven't personalized it, when we have the ability to understand things to the extent that we do, and we have the technology that we could apply, it just hasn't been applied to medicine. It's been applied to other things. It's sort of astounding that we're as late to the game as we are. Certainly other institutions have sort of picked this up, that, hey, wait a minute, we ought to be able to apply this technology and the technological development, the ability to handle big data to medicine. We're just clunky in the way we collect data and the way we apply care. So I think we're going to start to see a much more rapid change over time. So this is a great, it helps us to remind us that not every patient responds the same way to our treatment. We all know this, and then we just throw up our hands and say, oh, too bad. This guy didn't do well, and we did our best. Well, did we really do our best?
Go ahead.
Do you know to what extent these molecular variants have been tested or posited in animal models and in mice or any other type of models, and how they've responded to ACTH or other challenges?
No, I don't think people have. I haven't seen anyone who has put them into a mouse model. It might be a good thing to look at, and but I don't think they've done it. Most of the endocrinology work has been done, and they found, you find a mutation, and you find you find a sick person who's got some weird anomaly, and you test them. The problem is that we're testing more in trauma patients who are relatively normal, except for all the methamphetamine and alcohol. But it's like, but I think also the variability in response to stress is what leads to it. But yeah, it's a good idea. Maybe we could take a specific mutation, knock it up into a mouse or something like that. It might be worth talking about. Yes, sir.
If you do that, don't you have to consider the context? Because it's a lot more than just the gene. You have each of proteins, you have different chromosome subunits, you have all sorts of expression.
Absolutely. Yeah, I know. I agree, and it depends on what question you're asked related to it. But that's the issue: is that it is so complicated. So many things influence what happens. And we actually wrote a paper once that showed that a knockout mouse. Well, one reason we started doing the glucocorticoid receptor changes is that we thought a knockout mouse for the CD14 was just that knockout mouse, but we actually found that there was variability in the glucocorticoid receptor that was tied to it. So that's how we got down in some of that area. So yeah, those models help answer some questions, but not all the questions. But
we certainly are discovering that there are certain genes that influence
a lot more diseases than we thought. So the classic ones are the RET mutations, the RASopathies. So you are correct that our genetic medicine used to be, this is a funny-looking person, they must have a gene mutation. And now we're able to track the gene mutations first and say, gosh, maybe that is what's explaining some of these other diseases that we didn't know. And I think we're just starting to get better at that.
Well, and there have actually been pretty good studies where they, I can never remember how they say it, I think some analyses where they can take everybody and they do sequencing and look for polymorphisms for, all right, I'm going to look at all you guys, what polymorphisms you need. And they actually can do massive numbers of people and thousands of people. And here's the high frequency mutations that people have and which ones are of value. And so they've done a fair amount, but the problem what we deal with is it's the how do you handle stress, and that's hard to handle as far as it's not obvious because we don't know who's going to get burned today, and we don't know who's going to get in trauma today. So but hopefully we'll find more as we start digging through all our burn patients that we've collected this data.
Great.
All right, thank you.